Good morning, students. I'm Dr. Divya. Today's topic is Anthoceros. This is a systematic position of Anthoceros, division, bryophyta, class, Antheropsiptida, order, Anthocerotales, family, Anthocerotaceae, genus, Anthoceros. So, habitat. This distributed it worldwide, grows in shady and moist area of tropical and temperate region, about 200 species, and in Pakistan 3 common species are found in Himalayas: Anthoceros himalayensis, Anthoceros erectus, Anthoceros chamberlaensis. Vegetative morphology. So thallus, the plant body is gametophyte and consists of small dark green prostrate thallus. It is a rosette-like and with the lobes of whose margins are divided into small lobed segment. Dichotomous system is present. Surface of thallus is smooth, velvety and contains ridges and spines. Numerous thread-like rhizoids are present on ventral surface of thallus. So this figure shows thallus with sporangia of Anthoceros punctatus, and B figure shows thallus with sporangia of Anthoceros laevis. This is the thallus structure of Anthoceros. Mature sporangium splits open, releasing dead spores. So internal structure of thallus. First, there is a no internal differentiation. Parenchymatous cells are present. Each contain single lens-shaped chloroplast. Deep shells contain 8 chloroplasts. Each chloroplast contains single pyrenoid, and there are two surface, upper and lower epidermis. So lower epidermis contains mucilaginous cavity that open through a pore called slime pore. In these cavities, blue-green alga resides. So this is the lower epidermis or upper epidermis of Anthoceros. Thallus. Mucilage clapped, chloroplast, nucleus pyrenoid. In between that, alga filament are also present in thallus of Anthoceros. Reproduction is present in Anthoceros is performed by vegetative reproduction and sexual reproduction. So first vegetative reproductions, first method is death of older plant. Vegetative reproduction take place by the death of older plant, younger parts form new thallus, and second one is tubers. Some thallus forms tubers, so these tubers are rich in stored fat and protein, and these tubers germinate to on the margin of the lobes, so they can survive long period of drought. Tuber detach and form new plant. So vegetative reproduction is gamma. So gamma are also produced on stalk on the upper surface of thallus. These are also act as a vegetative reproductive bodies. Persistent apices. So in some species the thallus dies except the apices. They are called persistent apices. So they remain buried in soil and develop into a new plant body favorable condition. So this figure shows thallus with gamma cup. In sexual reproductions by Antheridia and Archegonia. So first Antheridia develops own cavity called antheridial chamber present on the dorsal surface. 1 to 25 Antheridia may develop in each antheridial chamber. So mature antheridium consists of an ovoid body with stalk. So there is a mass of spermatogenous cells surrounded by a jacket of sterile cells. So this figure shows antheridial chamber, a cluster showing a cluster of antheridia. So antheridia anthoceros that are male sex organ, and in A and B section, in A B figure shows stalk mature antheridia of Anthoceros crispus and lavis respectively, and C figure shows young and mature antheridia inside the antheridial chamber. Secondary bulbed antheridia are seen on the stalks of primary antheridia. So these are type biflagellated spermatozoids releases from antheridia. So dehiscence is like at maturity, the roof of the antheridial chamber rupture, exposing the antheridia, the apical cell of the uh, antheridial wall on absorbing water, rupture by apical aperture. So the antherizoids are now liberated to the covering film of water, and the antherizoids is spindle-like and biciliate. So the cilia are attached to the anterior end to the body, sometimes just near the attaching point to the flagella to the body. So the antherizoids swim in the water by the lashing movement of their flagella, archegonium. So archegonia are produced close to the growing point. So archegonia are embedded in the tissue of the thallus. Each archegonium consists of an egg and ventral canal cell, force canal cell. The canal of the archegonium is closed and the top by four cover cell. So these cells project slightly above the general surface of the thallus. Development of archegonium. This figure shows mature archegonium with mucilaginous wound fertilization. So what is essential for fertilization in the mature archegonium? The ventral canal cell, neck canal cell disintegrated and form a mucilaginous mass. It absorbs water, swell up and become out of the archegonial neck by pushing the cover cell apart. So these mucilaginous mass become continuous with the mucilaginous mound in the way in open passes down to egg is formed. Fertilization. So the mucilaginous mass consists of chemical substances. Many anthrozoids caught in the mucilage enter in the archegonial neck because of the chemotactic response. This is up to the egg and fertilization is effected. So prior to fertilization, egg enlarge and fills the cavity of the venter. Fusion of both male and female nuclei result in the formation of diploid zygote या oospore. Fertilization ends the gametophytic phase. Sporophytes. So the sporophyte of Anthoceros has certain unique feature. Sporogonium is born on the gametophyte, but mature sporogonium does not totally dependent on the gametophyte, and the mature sporophyte consists a bulbous foot and smooth, slender, erect cylindrical structure called capsule. Capsule varies in length 2 and 215 centimeter in different species. The sporogonium appears like a bristles or horn-like, hence the species are called hornworts. So it has following part food seta capsule. Structure of sporophytes, the food. So it is the basal part of the sporophyte, which is rounded bulbous structure deeply embedded in the tissue of the thallus, and the lowermost cell of the food are hystoria, which absorb water and mineral nutrient from the gametophyte for the developing sporophyte. Meristematic zone. So the intermediate meristematic zone, it is a narrow zone of meristematic cells located in between the food and the capsule. It regenerates the capsule from the base, thus the capsule are always in different stages of growth. Capsule. So the capsule forms the meson and conspicuous water of part of the sporophyte. It is a slender, smooth, upright cylindrical structure that slight tapers at the apex. It consists of capsule wall, sporogenous tissue, and columella. Capsule wall. The capsule wall is made of 4-6 layers of parenchymatous cells. The cells of outermost layer, which forms the epidermis, are heavily cutinized, vertically elongated, and interrupted by the stomata. Below the epidermal layer in the green parenchymatous photosynthetic tissue containing chloroplast. Thus, the sporophyte is capable of manufacturing their own food by photosynthesis. Sporogenous tissues. The sporogenous tissues of Anthoceros, so situated in between the jacket and the columella, at maturity it differentiates into spore mother cell and pseudoelaters. So columella basically is the central part of capsule is columella. It contains 4 rows of elongated vertical cell, but later it becomes 16 rows of cells. The transverse sections the columella appears to be a solid square, and the columella provides mechanical support to the capsule. So it also helps the spores to disperse and associated with the conduction of water and minerals. Dehiscence of the capsule. So at maturity, the tip of the capsule lose water and the loss of water from the capsule wall greatly favor in dehiscence. After dying of the capsule, a slit appears just below the tip before which gradually widens downwards. Eventually, the capsule wall split into 2 and 2 4 valves, and the hygroscopic to absorb moisture movement of the pseudolater is release the spore to a distance by air current at this stage.